Greetings again. This is Mike Jones, Biology 121, AP1, Anatomy and Physiology 1. We're continuing on with the early parts of anatomy and physiology. This time we're going to get into cells. Cells are the basic unit of life. I'm going to start this slideshow. You have the presentation available to you. Let me put it on full screen, so you can see both me and the show. I'll put me down here. Right, so the cells are basic unit of life. Cells are a compacted unit that all of the essential functions of life are done within that cell. That means there's a lot of structures inside the cell that help it to perform those functions, and each of these structures inside are specific. In fact, the cell has its own anatomy, and we have to talk about that. Cells are small. Now, cells can vary in size a fair amount. Red blood cell is about 7.5 micrometers, which is a millionth of a meter, on up to almost a millimeter for an egg cell, which is a pretty big cell. But most cells are, on the micrometer scale. They're fairly small. Obviously, you need a microscope to see them. Cells differentiation simply means that the cell has a specialized function. Of course, the original cell is the egg and sperm unite. That becomes one single cell, and from that single cell comes an entire organism. A dog, a cat, a bat, you. That can only occur because those cells become specialized. Each set of cells in a particular part of the body specialize in function and form. Their form, their shape, will determine what they can do. Now, all cells have certain basic internal components, and so we need to learn what those components are. So we're going to learn about a generic cell. What do cells have inside them? All right, let me get a pointer. Oopsie, no, that's not what I wanted. Technical difficulties. Here we go. This is what I wanted. And this is the other thing I want, the laser. Yowza. All right. Yeah, so cells are the basic units, right? But they come, look at these different shapes, different sizes, different appendages, different weird looking things. Sometimes they're long and skinny. Sometimes they're big and round and fat. Sometimes they look like pancakes. They're flat. Other times they have long projections on them. And sometimes the cells merge together so that they can work and help each other together. So we're going to look at a general cell. A composite cell. All cells have a nucleus. Let me amend that. Almost all cells have a nucleus. Red blood cells do not have a nucleus. Other than that, every other cell has a nucleus. A nucleus is where the DNA is housed, and it is kept separate from the rest of the cell. We're going to talk about what DNA does, how it works. Cytoplasm is the fluid portion inside the cell membrane. So cytoplasm is the fluid stuff inside there. There's a lot of components inside that. We talked about the organic and biochemistry of that last time. All of those organic and biochemical things are found inside cytoplasm. The cytoplasm is held within the boundaries of the cell membrane, and the cell membrane has its own special features to it. Right, so inside the cytoplasm, you're going to find organelles, specific functions, and the liquid itself, the liquid inside a cell, inside the cytoplasm, is called cytosol. So the cytoplasm is cytosol plus all of the internal components. Cytosol is the fluid or the liquidy portion inside the cell. All right, so here's a composite cell. This cell, you'll never find, this is not an actual cell. You'll never find all of these things that you see here. You're not going to find them in one single cell. However, these are all the things that are found in every cell, somewhere in the body. In particular, this thing back here called a flagellum, there is one cell that has one mammalian cell, maybe I should say that, one mammalian cell that has this structure. It is a sperm. A mature sperm cell has a flagellum. There's no other mammalian cell that has a flagellum. You can go through all the structures in here. We're going to talk about all these structures individually. I just want to bring your attention to a couple of different things. Well, one is the nucleus. You can see that the nucleus has its own cell membrane. It is walled off from the rest of the cell for particular reasons, for special reasons. Cell membranes are 2 layers thick. We call it a phospholipid bilayer. And you can see that here when we magnify it, the phospholipid bilayer. We're going to talk about the structure of the phospholipid bilayer. I hope you review what a phospholipid is, and what a lipid is, and what phosphates are, and how these work together. We're going to talk about that. We're going to bring that back up. But remember, phospholipid bilayer, that's going to be true for the cell membrane. It's also going to be true for the nuclear membrane. And then you have all these other specialized functions and features and other things. These are all called organelles, right? You have organs inside you. Your cells have organs inside of them. They're called organelles. All right, so the outside, plasma membrane, the cell membrane. Remember, we're talking about animal cells here, humans in particular, of course. We have a cell membrane or a plasma membrane. Plants have a cell wall in addition to a cell membrane. We don't have cell walls. We just have cell membranes. That turns out that's enough. That's all we need. Of course, it defines the outside of the cell. It maintains the integrity of the cell. Of course, it keeps cell stuff inside and keeps non-cell stuff outside. It is a structure that is called selectively permeable. Selectively permeable simply means exactly what the term says. Some things are allowed in, some things are not allowed in. Some things have to be escorted in, some things are kept out. So it is selectively permeable. Your cell membrane, in combination with the stuff that's inside the cell membrane, get to decide what gets in, what goes out, how does it get in, how does it get out. Is selectively permeable. Signal transduction, it permits the cell to receive and respond to messages. So there are things that happen on the outside of the cell that the inside of the cell needs to know about. There are ways to conduct signals from outside to inside or inside to outside. And then the consistency of a cell membrane is lipids and proteins. There are proteins that float around inside the cell membrane. We're going to look at those. Lipids are the primary component of a cell membrane. Now, I just mentioned it's a phospholipid bilayer. Think of a cell membrane as a layer of fat or lipid or an oily substance, if you will, around the outside of the cytosol and the nucleus that's inside. Right? If you think of it that way, it will make much more sense as to what can get into a cell and what cannot get into a cell. And we're going to talk about that. So just think of it as an oily layer or a lipid or a fatty layer around the outside. That's how to think of a cell membrane. All right, so the structure, as I mentioned, it's a phospholipid bilayer. That means, again, as I mentioned, if you think of the outside cell membrane as an oily or a fatty layer around the outside, that will help you to imagine what can get in and what can get out. What I'm saying there is if a substance is soluble in oils or lipids or fats, then it can slide right through a cell membrane. It can go from outside to inside or inside to outside if it is fat soluble. If it is not fat soluble, meaning it's water soluble or aqueous, it's in an aqueous solution. Say something like sodium chloride. Sodium chloride are ions, right? So you take table salt and you dissolve it in water, and sodium comes apart from the chloride, right? And so the sodium has a charge, has a +1 charge, and the chloride has a -1 charge. Those things are charged. They're ions, right? We talked about that. Ions cannot easily pass through a membrane because they have a charge, and the membrane itself, it has no charge, and it is kind of an oily or a slick substance. And if you think of it that way, you think, well, this polar substance cannot get through the non-polar membrane, so it's kept out. Likewise, things that are inside the cell that have a charge to them, that are polar, stay inside. They can't get out because they can't go through the fatty layer of the lipid bilayer. So that's one of the ways in which the membrane separates the outside world from the inside world. That's what a cell is. A cell is a divider between the outside world and the inside world. That means you can make things different inside the cell than they are outside the cell. That's how you make life. You have to make it different. So, things that are water soluble cannot easily get through a cell membrane. Things that are lipid soluble or fat soluble can easily go through a cell membrane. So, we would say in that case it's impermeable to water soluble substances. So that's the way to summarize that statement. Now, cholesterol is a component of a lot of cell membranes. Cholesterol is fat soluble. In fact, cholesterol is the basis for a lot of fatty steroid molecules like estrogen and testosterone. Well, cholesterol is found inside the cell membrane because it's a fatty substance, and it can easily slide around the cell membrane. It allows the cell membrane a little bit more fluidity, a little bit more elasticity, because cholesterol's in it. Now, inside a membrane too are membrane proteins. These membrane proteins float inside the cell membrane itself, and they serve as, well, they serve many functions. They serve as ways to receive signals from the outside world and tell the cell that something's at the door. It's sort of like a doorbell, right? Insulin is a good example. Insulin is a protein that floats around the bloodstream, but it locks onto a protein that floats in the cell membrane, and it's basically a door knocker, right? It says, knock, 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 insulin here, insulin is here, insulin has arrived, insulin is telling you to do what insulin does. We'll get to that at a later time. But it acts, these proteins that are in the cell membrane act like bodyguards. They keep things out, or they allow certain things in, but other things not in. They act as doorbells or doormen, if you will, telling the inside of the cell, hey, our friend insulin has arrived at the door. What do you want me to do? Things like that. There are also some of these proteins in the cell membrane form pores or channels. Again, they're like bodyguards. They allow certain things in, but they don't allow other things in. They're usually quite specific about what they will let in, what they will not let in. All right, so here we go. So here's our generic cell, our composite cell, right? We hone in on just this little section of the cell membrane, we blow it up, and then we hone in on just this little tiny section of the cell membrane, we blow it up again, we make it big. This is what we have. We have a phospholipid bilayer. What does that mean? Well, we have a phospholipid is this little circular thing. That's the phosphate part of a phospholipid. But remember, the lipids are those long, fatty tails that hang off the phosphate portion. Right? So these tails point to the inside of the cell membrane. On the other side, the other layer, remember this is a bilayer, two layers. So one layer has the phosphates pointing to the outside world. The other layer of phosphates is pointing to the inside world, inside the cell. In between those two phosphates are these long fluidy lipid tails. This is what gives it a bilayer, the lipid bilayer. This is what gives it that lipid oily nature. This is the barrier that charged molecules, polar molecules, cannot get through. This is the specific reason why this membrane is built this way, to keep some things out and to allow other things in, but only the things in that you want to go in. So, there's the structure. The phosphates tend to be on the outside, either pointing to the outside world or pointing to the inside of the cell. In between are the two lipid tails on each phosphate. So each of these phosphates has two lipid tails. Because they have this back-to-back feature, we call that a lipid bilayer. Now, here's a stylized view. We've sort of taken a cross-section. We've kind of sliced through a membrane. And we say, here it is, all right? So we're looking at the phospholipid bilayer again. Here's the lipid tails inside the membrane. And to the outside of the membrane is the phosphate groups on literally the outside of the cell. And then on the inside of the cell, there are phosphate groups inside here. Now you notice these little purpley things. These purpley things are proteins that are embedded in the cell membrane itself. And they serve those functions as signal transmitters, as pores, as channels. They are made such that they are able to float around in these membranes. And some of these proteins will have, see these little green things on there? Those are sugar groups that have been added onto. We call those glycoproteins. Glycoprotein is a sugar molecule added onto a protein. Glycolipids is a sugar molecule added onto a lipid, a special kind of lipid that floats around in the cell membrane. So you can see what these things are like. They're like little antennae. Right? They're floating around, they're antennae. They have a certain shape. And when something that floats through the bloodstream comes in contact with one of these things that is the right shape, it will attach to it. And it, as I said before, it acts as a doorman, or it acts as a doorbell, or it's telling the inside of the cell something has arrived. Now, the interesting thing is that this is how lots of things get into your cells. This is also how the virus that causes COVID-19 gets into your cells too. There's a special receptor on the outside of some of your cells that spike protein around the outside of the COVID virus attaches to something like one of these things. And when it finds that special connection, that virus now clamps on to that glycoprotein, and that allows the virus to enter the cell. So that's a clever little thing that, and a lot of viruses do this. I mean, SARS-CoV-2, that's the virus that causes COVID-19. It's not special in that way. All viruses, all bacteria that can enter into a cell, that can infect you, have some sort of special mechanism like that, where they can act like another molecule in your body, and they can specifically connect and gain entrance to your cells. That's how they do it. They do it this way. All right, now, there are these things called cellular adhesion molecules. This is very, very common in the immune system. Cellular adhesion molecules simply means that there are special molecules, proteins, say, on the outside of cells that allow certain molecules to recognize, let me give you this specific example here. This person, this is a person with a blood vessel, this is a blood vessel, and they've gotten a splinter. We've all gotten a splinter, right? You get it in your finger and you're like, ow! Well, if we magnify that view and we say, well, here's the splinter, here's what it's doing. It's jamming into a blood vessel, but it's creating a hole in that blood vessel. That allows certain molecules to be expressed on the outside of the blood vessels, of this blood vessel. This is a capillary. Those are called cell adhesion molecules. Now, there are special kinds. Some are called selectins, some are called integrins. I'm not concerned that you know that. When you breach that blood vessel because of the splinter, the cell membrane can change and expose these cellular adhesion molecules, to which white blood cells can now, that's their signal. That's their signal to say, something's happened here, you need to investigate. That cell adhesion molecule will now be expressed in this wounded area, and white blood cells will now invade that area looking for foreign invaders. If the splinter is not there, these cell adhesion molecules are not expressed on the outside of a cell membrane, and so the white blood cell simply goes right on past. So this is how your white blood cells know that something has gone wrong. These cell adhesion molecules are normally not exposed, they're normally not expressed. When you have an injury, they are expressed, they are exposed, and so the white blood cells now know, here's a problem. All right, let's talk about cytoplasm. So cytoplasm is the fluid. So here you go. Cytoplasm is cytosol. Remember, cytosol is the fluid itself, and then all of the organelles that are inside the cell membrane. When you put the organelles and the cytosol together, we have this thing called cytoplasm. And so here's the definition. The cytosol is the fluid part, and the organelles are the structures. Now, we say solid structures. Well, they still have little membranes around them too, and they're kind of fluidy and they float around. But yes, they're certainly more solid than the fluid that they're floating in. Now, all cells in the body have a cytoskeleton. Just like you have a skeleton that gives you shape and form, so do cells. That's why you can have certain cells that are flat, certain cells that look like cubes, some cells that look like tall columns. It's because they have a cytoskeleton that the rest of the cell is kind of built around, if you will. All right, now we're going to talk about organelles. So what are these organelles? There are some organelles that you really need to know about. Very important, ribosomes. Ribosomes are, they come in two forms. They can be free-floating in the cytoplasm. They can float around. A ribosome is a protein, or a protein complex. Maybe that's a better way to say it. A protein complex it also has RNA as part of this protein complex. What ribosomes do is they are the factories that make proteins. They get their instructions to make those proteins from mRNA. So we'll talk about that later on. But mRNA tells the ribosome what order to put the amino acids in on the protein that it's going to make. But it's the ribosome that does that. So ribosomes can be floating around in the cytoplasm, or they can be attached to another component of the cell called the endoplasmic reticulum. And the plasmic reticulum are membrane bounds. They're like these flat membranes, all smushed up against each other in many rows. If a ribosome is attached to it, it can make a protein against that endoplasmic reticulum. In that case, if a ribosome is attached to an endoplasmic reticulum, we call that a rough endoplasmic reticulum. If there is no ribosome, there are other parts of the endoplasmic reticulum that do not have ribosomes attached to it, and so we just call that smooth endoplasmic reticulum. We call it rough because if you look, like especially here on this micrograph here, this is a really good micrograph. If you look very closely, you'll see these flat areas, these flat walls. Now that is another lipid bilayer. That is the endoplasmic reticulum. On this particular endoplasmic reticulum are these bumpy things. Those bumpy things are ribosomes that are attached right onto the endoplasmic reticulum. So that means that proteins are being manufactured right here by those ribosomes, and those proteins are being packaged inside the endoplasmic reticulum. All right, more organelles. Vesicles. You're going to see lots of kind of vesicles. A vesicle is anything that's inside a cell that has a small spherical membrane around it. Now, inside that vesicle, different kinds of things are happening. We're going to talk about the kinds of things that you might find inside those small vesicles. They're just membranous sacs, right? They're little sacs. They're like little balloons, but they have special things inside them. So sometimes they will have enzymes inside them that will destroy, say, bacteria. If a bacterium has invaded this cell, this vesicle may have bacterial destroying enzymes inside it. There are other things that the vesicles can contain as well. They can store certain products or compounds maybe that particular cell needs. They're very, very useful, but there's something special that's inside that vesicle that cell needs. Next organelle is called a Golgi apparatus. Now, a Golgi apparatus these are sacs of flattened membranous discs. They are pretty much a continuation. You can think of them as a continuation of the endoplasmic reticulum, right? So the endoplasmic reticulum may make a protein. It may have the protein inside the endoplasmic reticulum once it's all made, and then as that protein will travel out in the endoplasmic reticulum, the Golgi apparatus will help to package that protein. So a cell, an example of that would be a cell in your pancreas that makes insulin. Insulin is a protein that has to be made in a cell in your pancreas, but then it has to be put into the bloodstream to have its effect throughout the body. It would be manufactured by a ribosome on an endoplasmic reticulum, and then it would be transported into the Golgi apparatus. The Golgi apparatus would then wrap it up into a vesicle, in a sense, a vesicle where the insulin is inside, and then that would be sent outside the cell and put into the bloodstream. Here's another example of where you would get a vesicle. Milk secretion is an example of an organelle interaction and the transport of substances by vesicles. It's the same thing as my insulin example. This is just with milk, right? So a special cells would make breast milk. That milk is packaged. It's made in the rough endoplasmic reticulum. It is packaged into Golgi apparatus. It is then that becomes a vesicle where the milk, this is the milk protein inside the vesicle. The vesicle then is shipped out the cell. And there are special processes by which that milk is expressed. But these organelles, that's what the organelles are designed to do. That's what they do in a milk-producing cell. They do different things in an insulin-producing cell or a thyroid hormone-producing cell. I mean, on and on and on. All right. Now, an extremely important organelle. Every cell has this. Mitochondria. Mitochondria, you've heard it. I'm sure you've heard it. You've heard it in high school biology. These are the powerhouses of the cell. These are the energy manufacturing stations. Energy in the body is ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. That is energy. That is cell money. That's how things get done in a cell. You need energy to power all of these processes. Mitochondria produce that ATP in huge quantities. So that's what a mitochondria is for. That's all mitochondria do. They also have, you can see, here's our stylized cell again, right? We blow it up, we look at this one component. Here is a mitochondria. What you can see about a mitochondria is there are lots and lots of membranes on the inside. And it turns out that really what it is there's a membrane around the outside of a mitochondria. Then there is an inner membrane. That inner membrane is very curvy, very, very circuitous. Inside that inner membrane is where a lot of the ATP processes are happening. We call that inside, that the space in between the inner membrane, we call that the matrix. Not the matrix as in the movie The Matrix, but the matrix as in where energy is produced in your body. ATP. We're going to talk about that too. All right. Now, these are more specialized organelles. Lysosomes and peroxisomes. These are, as you can see, they are like vesicles. They're vesicles. They're just sectioned off, balloon-shaped objects, I suppose you could say. Inside lysosomes are enzymes that digest bacterial proteins, or they digest bacterial nucleic acids, bacterial carbohydrates. They are areas where this is a good description, I think. The garbage disposals, the worn out parts of the cell. If they're no good anymore, if they need to be recycled, they're put into a lysosome. They're broken down to their component pieces, and they can be used again. Peroxisomes are quite similar. Again, they will try to digest lipids, alcohols, hydrogen peroxide, but similar. They're very similar to lysosomes. So lysosomes and peroxisomes will have specialized enzymes inside their sacs. Now, you certainly wouldn't want these things to open up to the rest of the cell, otherwise you would digest the cell itself. So you got to keep these things walled off away from the rest of the cell. Right. Other stuff. Most of these things, microfilaments, microtubules, intermediate filaments, those are components that help to give the cell shape, right? This is the cytoskeleton. These are the components that help to maintain the shape and the sort of the skeletal structure of the cell, the shape of the cell. They do help some cells move. They also help to transport things around the cell itself. So they serve as kind of a scaffold. And so things that need to move through the cell can hang on to the cytoskeleton. They can hang on to the microfilaments and the microtubules, and they can move things around in the cell by, kind of like you could think of them as ropes or a scaffold on which these other things can move from one place to another place inside the cell. So now we're getting into stuff where these are the kinds of components that help a cell divide. And we're going to talk about mitosis in a bit. Centrosome. A centrosome has two centrioles, right? So a centriole, one centriole, or a centrosome by itself really doesn't do anything. But it becomes much more functional when this cell is going to divide. When the cell divides, then the centrosome will also replicate itself first. And so then you will have two centrosomes. It is the centrosomes that coordinate these long spindles on which the chromosomes will hang on just before the chromosomes are pulled to opposite ends of that new cell, so that you have the same number of chromosomes in one cell as you have in the other cell. It's the centrosome that coordinates that pulling action. Right, so it says they produce spindle fibers during cell division. We'll talk about how they do that. Cilia. Some cells have cilia. In particular, cells of the respiratory tract. as it says, they form a fringe. Cilia do have the ability to move. They can move themselves. They have kind of a muscular protein inside them that allows them to move in a coordinated fashion. The point to having cilia is, especially in the respiratory system, if you're breathing in and you're breathing in dirt particles and dust particles and you don't even know it, and those dirt and dust particles will get caught on the cilia. Now these cilia also have a mucus, a layer of mucus on top of it. So this dust and dirt and stuff will get caught on the mucus that is on top of the cilia. Now the cilia will work to remove those dust and dirt particles out of the body in this coordinated rhythmic movement fashion. And so that's why you have to blow your nose. That's why your nose drips and runs. And that's why when you have a respiratory infection, or even if you have sinusitis, I have chronic sinusitis, not that you wanted to know that, but you'll hear me do that, and I will swallow the mucusy stuff. This is more detail than you ever wanted, I know that. But it's the cilia that will keep dirt and dust particles out of my lungs and transport it outside the body or into my gastrointestinal tract, in which case I'll swallow it then, and then my stomach acids will destroy whatever's in there. It's also propelling the egg towards the uterus. In the fallopian tube, or in the uterine tube, you're going to see there are cilia there as well that move the egg along. Flagella. As I said, sperm. A sperm has one flagellum. A flagellum is a tail. It's a propulsive tail. Next, nucleus. As I said, all cells have a nucleus, with the exception of red blood cells. They do not have a nucleus. The nucleus houses DNA. Now, DNA in most cells that are not replicating is held in the form of what we call chromatin. Chromosomes are the condensed form of chromatin. When a cell is going to replicate, it takes all of the DNA and packages it up into a very, very tight structure. And that tight structure is called a chromosome. But when it's not dividing, when you don't need it to be compacted like that, it just free floats, and it's these long strands of DNA. And those long strands of DNA are called chromatin. And they float around inside the nucleus. And the outer membrane of the nucleus keeps it separated away from the rest of the cell. There is a particularly dense thing inside the nucleus called a nucleolus. A nucleolus is a dense body of RNA and protein, and it is the site of ribosome production. So ribosomes are made inside the nucleus, and then they have to go outside the nucleus to do their work. Right? We already talked about ribosomes. They can float around the cytosol, or they can be attached to endoplasmic reticulum, and we call that rough endoplasmic reticulum. So they're made inside the nucleus, in the nucleolus, and then they're shipped outside the nucleus, outside the nuclear pore, or outside the nuclear membrane, through a hole in the membrane. And a hole in a nuclear membrane is called a nuclear pore. Now, the pores allow some things to leave the nucleus. They don't allow the DNA to leave the nucleus. They're bodyguards there that say, whoa, 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 whoa. Sorry, DNA, you got to stay here. Oh, ribosome? Go ahead. Come on through. Oh, mRNA? You betcha. Come on through. Yep. So there are guards there, too. Bodyguards. All right. Movements in and out of a cell. Stuff has to get into a cell. Stuff has to get out of a cell. You have different ways of doing that. There are what we call passive processes. Passive means there's no energy. You don't need to expend any energy. It's a natural process. it's going to take place without the use of any energy at all. Now, these tend to be processes that are similar to chemical processes, right? So the first one there, first passive process that you see there is called diffusion, right? This one called diffusion. Diffusion is defined in chemistry. Diffusion is the movement of a substance from an area of higher concentration to an area of lower concentration. That's a passive process. We all know, you've seen this happen before. That's why you put cream in your coffee. Say you have coffee, right? You put cream in there, and if you just let it diffuse, it takes a while. It's kind of cool to watch the milk swirl around and stuff, and it will even itself out all through the coffee if you give it enough time to do that. But usually we don't have the time. We don't want to take the time. So what do? You stick a spoon in there, right? You spin it around. You are hurrying diffusion along. You're making diffusion go faster. But that's still diffusion. Diffusion takes no energy. You can just let it happen. So diffusion, by definition, also is the movement of a solute. So in any solution, when you, we'll take the coffee and cream example, okay? Coffee would be the solvent. Now, of course, it's water is really the solvent, but we're gonna take some liberties with this. If you have coffee, we're gonna call that the solvent. When you put something inside the coffee, let's say you put sugar in the coffee, right? You take a pack of sugar and you dump it in there. Well, that's a solid. It's going to dissolve in the solvent. So the solute is the thing that you are dissolving in the solvent. All right, so if it's coffee, you put sugar in there. If you just let it go and walk away from it, that sugar will dissolve. It will take some time to do it, but it will do it by diffusion. Passive process, you don't have to do anything. Those sugar crystals will dissolve in the water, in the coffee, and then they will spread themselves out evenly in all directions. All right, so that's diffusion. Diffusion refers to specifically the solute, the thing you are dissolving, spreading itself out and evenly dispersing itself inside the solvent. In biological systems, the solvent is always, are you listening to me? Always water. Water is always the solvent. Things dissolve in water. That's how biological systems work. You take organic chemistry and it doesn't work that way. There's other stuff. For physiology, it's always water. Okay? So, if something moves in water and dissolves in water and disperses itself evenly in water, that is called diffusion. However, you can think of it the other way. Well, water can also move. Water has to move around. It will become more clear what I mean when we start talking about cell membrane, when we bring cell membranes back into this conversation. But water can move from one place to another place as well. If water moves, it is called osmosis. So, in physiologic systems, if the solvent moves, which is always water, it's called osmosis. If the solute moves, the thing that's dissolved in the water, if that moves, that's called diffusion. You have to really understand these concepts because we're going to really get hot and heavy into this. We're going to take the deep dive into this. All right, so that's diffusion and osmosis. We're going to come back to those, don't worry. Facilitated diffusion is just a way to help diffusion move a little faster. We'll talk about specific ways in which that can happen. And then filtration is also a passive process that also does occur in physiologic systems. You're more used to it in chemistry lab, where you put a funnel and you fold up filter paper and you stick it in there, and then you pour some solution that has something dissolved in it, but not all of it's dissolved, or you're trying to get dirt out of it, or whatever you're trying to get out, right? That's called filtration. Well, it's a passive process because you're using the force of gravity, or you're using some sort of external pressure that already exists. So gravity is there. You pour this stuff into the funnel, you have your filter paper, and you wait. It's passive, right? You don't have to do anything. The force that causes the filtration is gravity. Gravity pulls the water through the filter. The stuff that can't go through the filter stays behind. That's called filtration. That's a passive process. There are several places in the body where that occurs. All right, so those are passive processes. Next up, we've got active processes. Active processes, they require ATP. When we talk about energy requiring processes, an active process, when I use this word active, and I will start to use it a lot, active transport, I'm going to say, and I'm going to use these other words. Whenever I use any of these words, you have to think this requires the input of energy. And the input of energy in physiologic systems means ATP. Always. Imprint it on your forehead, tattoo it on your arm. ATP. You've got to have ATP for active processes in the body. So these are the biggies, right? Active transport, endocytosis, exocytosis, and transcytosis. We're going to talk all about what these things are, but they are active processes. They require energy. The other ones do not require energy. All right. Now, I just went through diffusion. Here's a picture of diffusion. Remember, in physiologic systems, water is always, what did I say before? Water is always the solvent. It is the thing that other things are dissolved in. All right? So, we take, in this case, I don't know, we'll say it's a sugar cube. Oh, yeah, sugar cube, okay? So we take water, we dump a sugar cube in there. At first, it doesn't do anything. And then we give it some time. Time is the important factor for diffusion. The longer you wait, the more of those molecules will be dispersed in the water. And if you wait long enough, all of the sugar will be evenly dispersed all through the water. So, at first you have a high concentration of sugar right here. And then it starts to dissolve. You still have higher concentration of sugar here than you have in other places. But slowly but surely, these sugar molecules dissolve in the water and they disperse themselves as evenly as possible all through the container that they have. Right? It's a passive process, no ATP. Now, in this case, if we were to think of this beaker as a cell membrane, we would say this sugar will spread itself out evenly throughout the container of water within this cell membrane, within the beaker. And so that's diffusion. Now we're going to change the situation a little bit, okay? We are going to, in this case, we are going to introduce something in between or in the middle of our beaker. So we would say that the walls of the beaker, of course, are impermeable, right? They don't let anything through. Water, sugar, whatever you're dissolving in the water. The beaker itself is an impermeable barrier. Nothing can get through there. Great. But in this particular case, what we're going to do is we're going to insert this particular membrane. Right? And so this membrane, we're going to say that this membrane is semi-permeable. It is not, it is selectively permeable. And we're going to tell you what it it is selectively permeable to. So, and that's an important thing. I guarantee you, I will give you a test question that will test your ability to read the question, because I'm going to tell you here, the very first thing it says, the membrane is permeable to what? To both water and solute. Okay. It is permeable. I'm telling you that. This example is telling you that. It's permeable to both water and solute. Okay. So pretty much as far as you're concerned, for our example here, this is not a barrier. It's permeable to both things that are inside the beaker. So guess what? Both things inside the beaker will spread themselves out evenly. This situation that you're looking at right now is not different than this situation. Here there is no barrier, and so the sugar disperses itself evenly throughout the beaker, because that's diffusion, because diffusion requires that it spreads out all over the place, right? Here, yes, I can tell you I'm putting a semi-permeable membrane, but my membrane here in this particular example is permeable to everything that's inside my beaker. Well, it's as if there's nothing there. So that means that the solute, which is dissolved, and you can imagine that it's sugar, just like in the last example. Right, here's the solute. It's dissolved in the water. It's permeable. The membrane is permeable. So it's as if the membrane wasn't there. So it goes through the membrane, and it evenly distributes itself. If you give it enough time. Give it enough time, it evenly distributes itself. The other way that you have to think about this too is that it is permeable also to water. So that means, see, this solute over here takes up some space that would normally be taken up by water. So in the beginning, there's a lot of solute on this side, and there's a little bit less water, which is a weird thing to think. There's a little less water on this side than there is on the other side. So the solute wants to evenly distribute itself, and it does. But so does the solvent. Water also wants to evenly distribute itself. So in the end, you wait it long enough, you're going to get an even distribution of water and solute, sugar, if we say it's sugar. All right. Once you hit this point here, this is called equilibrium. Now, again, I'm going to reiterate, because this is going to be a test question, and I guarantee it. This statement is important. Membrane is permeable to both water and solute. Now I'm going to change the situation. So now we're going to change it. Well, we'll get to the change in a minute. Facilitated diffusion. So that's diffusion. That's also refers to osmosis, because water is moving through that membrane. So that's osmosis. Let me say that one more. Let me say one more thing about this situation. The solute goes across the membrane and distributes itself evenly on both sides, and water molecules will also evenly distribute themselves on both sides of this membrane. So what you have here is both. You have both diffusion. Diffusion refers to the solute, and you have osmosis, because water is also moving to evenly distribute itself. So you have, in this example, this slide really should say diffusion and osmosis in this particular example. All right, now we move on. Facilitated diffusion means that we have set up a concentration gradient. Concentration gradient simply means I've got more stuff, more solute on one side of my membrane, of my semi-permeable membrane, than I have on the other side of my semi-permeable membrane. We call that a concentration gradient. Right? So diffusion wants to happen. Diffusion wants to occur. But remember, I said So this is a really good example. Water-soluble substances. But remember, I told you that water-soluble substances cannot go through a lipid bilayer membrane on their own. They can't just simply slide through that membrane. They have to be allowed to go through the membrane, and they're allowed to go through a channel or a pore, whatever this thing might be called. And it's a protein. So this is a protein that is embedded, as I showed you before, it's embedded inside the cell membrane. And this situation is such that the cell has made a higher concentration of whatever it is outside the cell. Sodium's a good example. If I put a lot of sodium on the outside of the cell, the ion sodium, if I have a pore, a particular protein that allows sodium to go through, well, then sodium will go through. This is called facilitated diffusion because it's still diffusion. It's still, sodium is moving from a high concentration to a lower concentration. But normally it can't do it through a cell membrane. If I stick a pore in there that is specific for sodium, then the sodium will go down its concentration gradient. If it's allowed to do it, it will. Right? So I'm helping diffusion along. It's facilitated diffusion. I'm helping diffusion along. But it still doesn't require any energy. I don't have to use any ATP to make this happen. All right. Now we're going to come on to osmosis. Now in this case, so again, it's really, really important that you read this. Osmosis is the movement of water across a barrier, across a selectively permeable membrane, from a region of higher water concentration to a region of lower water concentration. Which this is a statement. I say it every semester. This is a statement that kind of blows people's minds, because you start thinking, what do you mean the movement of water? We're used to thinking of diffusion. We're used to thinking of the things that are dissolved in the water moving around, but we're not used to thinking of the water moving around. Well, osmosis is, by definition, the movement of water. If water can move, it will move. But water wants to even its concentration out, as well as the solute wants to even its concentration out. Everything wants to come to equilibrium, and everything wants to be even Steven, same concentration. Everything is dispersed exactly evenly all the time, everywhere. That's how chemistry wants to work. But physiology requires that we separate things, that we make different concentrations of stuff inside the cell, outside the cell, across a semi-permeable, a selectively semi-permeable membrane. All right, now we're saying in this time, you see here the protein molecule, we have a solution here, and we have a protein molecule dissolved on one side of the membrane, okay? We have, I wish they had colored this differently. What we have is we have a protein molecule on one side, and in this example, the protein. Let's say you could say it's anything. It doesn't matter what it is over here. You could say it's salt. You could say that it's sugar. It can be protein, as they say here. Okay, but they're telling you this is a selectively permeable membrane, and protein cannot cross the membrane. So if protein is on one side, if protein's on one side of this membrane, it can't go through to the other side. It might want to. It wants to naturally diffuse over to the other side of the membrane, but it can't do it. So it has to stay on its side of the membrane. All right? But the water is evenly distributed, as we say. So let me set this scenario up better for you. We have a beaker of water, okay? Just plain old water. Before we got to step 1 here, we have a plain old beaker of water. We introduce a selectively permeable membrane in the middle of the beaker. Okay, so I stick that in there. Now I'm telling you that this protein or sugar or salt or whatever it is cannot get through the membrane. Okay, so now I'm going to put that protein, sugar, salt, whatever, on one side of that membrane. Okay? It can't go through the membrane. Okay? The only thing that can move here is water. Well, what water wants to do is it wants to dilute, I say it wants, right? It's like it has a mind, like a brain, like it's thinking about it. Well, what it wants to do is it wants to dilute that protein, sugar, salt, whatever that's on the other side of the membrane. It wants to dilute it so that the concentration of that solute would be the same all over the beaker. But it can't be the same all over the beaker. It just can't be. So the water will move over to where, what you have to think of is over here, if I have introduced this protein, salt, sugar, whatever's over there, if I've introduced that, what I have just done is that stuff is taking up some of the space that used to be occupied by water alone. Well, the water on the other side wants to help out, I guess, in a sense, if we want to continue with my, water's thinking about this. The water will then move across that membrane over to the other side. And it will try to dilute the other side. It'll try to even things out. But it can only even it out to a certain extent. What stops it is eventually, see this column of water, right? So now the water levels were even on each side of the membrane when we first started. Now the water levels are not even. So what you have here is osmosis wants to take the water from the right side to the left side. But hydrostatic pressure, gravity here is pushing down. Gravity wants to push the water back to the other side. So you have to have a balancing act. Hydrostatic pressure balances against osmotic pressure. So we call this an osmotic gradient. And that an osmotic gradient creates what is called osmotic pressure. You need to understand this term because we're going to run into that in the very next thing when we start talking about different concentrations of solutions. All right? Osmotic gradient is the concentration of water from one side to the other side across the membrane, across the semi-permeable barrier. If water can move, it will move. If the solute can move, it will move. If the solute cannot move, in this example, it cannot. The solute can't move, so water can move, so water does move. But it can only move as far as until it creates an equilibrium with hydrostatic pressure. This is a natural process. It does not require energy. You don't have to make this happen. It happens all by itself. This is passive. Now, I told you, here we go. Osmotic pressure. That is exactly what we talked about here. This is osmotic pressure. Osmotic pressure wants to bring water from the right to the left. But gravity creates what we call hydrostatic pressure, and that wants to push down on this water and shove it over to the other side. Eventually, these two forces balance each other out. When they balance, you get the beaker on the right. Now let's talk about cells. The same thing happens in cells. If you have a water concentration, so these are red blood cells. You'll do this in lab. You're going to see this in lab, right? These are red blood cells. Okay, this is a normal red blood cell, and this red blood cell is in a solution that has the same concentration of water and salts on the outside as it does on the inside. That solution is called an isotonic solution. An isotonic solution means that the water and the solutes that are outside are the same as those that are on the inside. Everything is balanced. Then you get a normal red blood cell. That's A. Okay? Hypertonic. A hypertonic solution means the outside of the cell has more salt in it Usually we're talking about salt, so we're going to talk about salt here. It is more salty. It is saltier on the outside. What does that mean? It has a higher concentration of salt, but that also means it has a lower concentration of water. If there is more solute, there is less water, which is a weird thing to think. But if you think about it that way, it will make more sense. So more salt on the outside, less water on the outside. That means on the inside, there is less salt and more water. So what the water wants to do, the water wants to move. The salt cannot move. This is a semi-permeable membrane. Remember I said that things that have a charge to them or they have a polar attribute to them chemically, they cannot get through a cell membrane. If it cannot get through the cell membrane, it can't move. It can't go from outside to inside. It has to stay there. So this higher salt concentration stays on the outside. The water can move. The water that is on the inside of this cell wants to move out to dilute that higher concentration of salt on the outside. And so it does. It moves out. As it moves out, the water leaves the cell, and of course the cell starts to shrink down. Less and less and less water. This prickly shape that you see there, that is what the cytoskeleton looks like. That's what the skeleton of a red blood cell looks like. So this is like the skin of that red blood cell is sucking down onto its skeleton because the water is leaving the cell. We call this a crenated cell. C-R-E-N-A-T-E-D. Crenated. This is a crenated red blood cell because it is in a hypertonic solution. Osmotic pressure causes water to leave the cell. All right. How about the other way? The other I got to move myself so you can see this cell. All right. Hypotonic means there is less salt on the outside of this cell than there is on the inside of this cell. Less salt. Hypotonic. Less salt on the outside. And when we're talking about isotonic, hypertonic, and hypotonic, we are talking about the solution that the red blood cell is in, not what's inside the cell, what is outside the cell. Hypotonic solution means there's lower salt outside this cell. What does that also mean? Lower salt means more water. There's a higher water concentration outside this cell than there is inside the cell. Water can move through the membrane. It wants to move. It will move. It's going to move into the cell. It moves into the cell, into the cell, into the cell, and this cell is now blowing up like a balloon. We're shoving water inside there. That red blood cell gets bigger and bigger. So normal red blood cell here, big, full of water red blood cell here. This cell is losing water. Hypertonic solution gives you a crenated red blood cell. A hypotonic solution gives you a big spherical red blood cell. We can take an extreme example. If there were no salt in this water, if it were just water, I just put pure water on the outside of this red blood cell, there's no salt at all, then that would be like the extreme example of a hypotonic solution. All of that water now will go flooding into the red blood cell, and it will keep flowing in until the cell membrane cannot handle all of that water, and that red blood cell will bust open. Now that's like if you were to take a balloon, you blow and blow and blow and blow, eventually that balloon pops. That's what happens to red blood cells. They pop. That's called lysis. That's called hemolysis or hemolysis. You are rupturing a red blood cell membrane. This is what would happen if you got an IV, an intravenous fluid was put into your veins of pure water. If you were to put pure water into your veins, you would start to lyse your red blood cells. This is why we don't use IV drips of pure water. We use IV drips of what's called normal saline. Normal saline is 0.9%. There's salt. It's salt water. The reason we have salt water is because we want an isotonic solution. We want an isotonic solution so your red blood cells are happy. If you put in a hypotonic solution, or in fact pure water, into your veins, you're gonna lyse your red blood cells. So it's always my recommendation that if you're in the hospital for whatever reason and they come up to you and they say, we're going to start an IV, and that IV, the bag says pure water, you should run away because they're trying to kill you. That would never happen, of course, obviously, but still. There you go. Okay, filtration is pressurized. This happens all the time in your blood. Things leave your blood, your vessels, your blood vessels, by pressure. They're pushed out. Yes, all the nutrients that need to get into your cells all over your body get that nutrition by filtration. And because your heart beats, your heart creates a pressure on the blood vessels, and that pressure pushes things out through. Your capillaries are slightly leaky, and they need to be leaky because your cells need nutrition that is coming through the capillary wall, and it comes through there by filtration. No energy required, no ATP required to do this. All right, now we get to the active mechanisms. Active transport is a huge one used all over the body for all kinds of stuff. Active transport, what you're trying to do with active transport is you're trying to move something against a concentration gradient, right? And we've talked. I talked a little bit, well, a few minutes ago about facilitated diffusion. Facilitated diffusion is where we allow things that are at a higher concentration, we allow them to go through the membrane to a lower concentration. How did we get that higher concentration on the outside of the cell? Haha, we got it by active transport. We created that imbalance of ions. It's a very, very common thing that your cells do. Nerve cells are famous for this. Nerve cells, pump ions, some ions, outside, out of the inside of the cell to the outside of the cell. So we're creating a high concentration outside the cell and a low concentration inside the cell. That is done by active transport. All right, active transport. So the biggest and best example of active transport, I mean, all of these are great examples, but the biggest and best is what's called the sodium-potassium pump. Sodium-potassium pump pumps sodium outside the cell and it pumps potassium inside the cell. So most cells have a high concentration of potassium inside and a high concentration of sodium outside. That happens because of active transport, because of the sodium-potassium pump. We have to use energy to do that because we are pumping these things against their concentration gradients. So you need energy. You need ATP for that to happen. All right, endocytosis. Now we're getting into bigger things. If we have to move bigger things in or out of a cell, in this particular case, endocytosis is moving something from the outside to the inside. There are different kinds of endocytosis, but you're basically taking something outside the cell and you're bringing it inside the cell. But you're doing it by kind of wrapping the cell membrane, part of the cell membrane, around the thing that you're bringing in. So if the thing that you're bringing in is a liquid, if you're bringing that liquid into the cell, that's called pinocytosis. Now, it is a form of endocytosis. Pinocytosis brings liquids into the cell. Phagocytosis brings solid particles into the cell, like a bacterium. If you were going to chew up a bacterium, you have white blood cells that want to eat up bacterium. How do they do it? They bring it into the cell by endocytosis. It takes energy to do that. You need ATP. Third way that it happens is what's called receptor-mediated endocytosis. Pinocytosis is just, pardon me, if you're bringing the fluid in, there's nothing special. You just sort of wrap the membrane around the fluid, whatever the fluid is. Could be water, could be other fluids, lipids, fats, things like that. You suck them up. Phagocytosis is you're taking a solid particle. You don't really particularly care what the solid particle is. You're just engulfing it, sucking into the cell. Receptor-mediated endocytosis is a more specific way. You're bringing a specific thing that you are looking for outside the cell. The cell is looking for a specific thing, and it has a receptor, one of those proteins that's floating around the cell membrane. This is how viruses get into your cells. You have this receptor on the outside of your cell membrane. That virus comes along, attaches to that receptor, and then this is called receptor-mediated endocytosis. The cell brings the virus in, and you want to tell the cell, what are you doing? Don't do that, you crazy cell. Come on, stop it. That's called receptor-mediated because it is a specific receptor that brings a specific thing into the cell. All of these things are endocytosis. They all take energy. So here's the examples. Right? If it's a cell, let's say it's a white blood cell, a particular white blood cell that wants to eat up bacteria. Okay? So it brings in the bacteria into the cell. It merges the bacteria with a lysosome. Remember we talked about lysosomes, and they have these enzymes that are going to chew that thing up, whatever it is, bacteria in this case. You merge the lysozyme with that phagocytized particle. You chew it up, the enzymes destroy whatever's in there, and then these products, these digested products from this cell, are spit out. Now, there's other things that happen to the digested products too, but this is one thing that can happen. This, on the bottom here, is receptor-mediated endocytosis, a specific thing, a specific molecule, or a virus in the case that I told you before, attaches to a specific receptor on the outside of the cell. Once that specific thing attaches to its receptor, that specific thing is now brought into the cell. In my example before, it's a virus. It doesn't have to be. It isn't normally a virus, but it can be a virus. But this is called receptor-mediated endocytosis. Now, just as you can bring things into the cell, you can spit them out. Exocytosis is the way that's done. You're releasing a particle, releasing neurotransmitters from nerve cells. This is a great example of exocytosis. These things, the neurotransmitters, are made inside a nerve cell. They're made either at the endoplasmic reticulum, they're transported through the Golgi body, they're made into a vesicle. So the neurotransmitter is inside this vesicle. The vesicle then merges with the cell membrane of the cell, and that stuff, in this case we'll say it's neurotransmitter, that neurotransmitter is released from the cell by exocytosis. This takes energy, ATP. Transcytosis is sort of a combo of endocytosis and exocytosis. You're endocytosing something on one side of the cell, you're transporting it through the cell, and then you're spitting it out on the other side of the cell. So transcytosis simply means whatever this thing is. They use the example of HIV, which is the human immunodeficiency virus. The disease that it causes is called AIDS, acquired immune deficiency syndrome. That virus travels across, as they say, the lining of the anus or the lining of the vagina, the epithelial lining. It's brought in on one side, endocytosis on one side, and exocytosis on the other side. That's called transcytosis. This requires energy. Hey, I am back in this slideshow. What I've decided to do here is to stop the slideshow. I don't want to get into the cell cycle just yet. I am going to end this particular video, and so the end of this, the end will be, where's the last slide? There's the last slide. We'll end with transcytosis, and this one. I will pick it up in another video with other topics. So I will see you on the other side with another video. Thanks for your attention. Appreciate it.